Hello guys, welcome back to Empower Tutorials. This is your master teacher, Dr. Sonam. So yes, guys, we are back on this amazing, online platform that we've got to get to know each other, to interact with each other. So let's keep this going and let's continue with our plant physiology session. Session 2 we are on, right? We have started with the session 1. We have completed session 1 and spoken about all the basic terminologies that we have seen in absorption of roots, right? So now we are going ahead and seeing all the other detailed theories of absorption of roots, right? So now let's go ahead. Yes. So before we move ahead, I had given you all one activity, okay, in which I asked you all to put a handful of raisins in water and leave it overnight and then check and tell me what happens to those raisins when you check them in the morning. So I am so delighted to receive so many answers from all my children, Deepak, Gunjan, Sahil, Simran. So many answers I've received. I'm really happy kids. You all have actually gone back home and done that experiment. So really good. Yes. So correct answers I have got from all of you. So yes, the name of that process is imbibition. Okay, उसमें अब क्या हो रहा है वो raisins, we have put those raisins in water, right? So now what has happened is those raisins which are actually shrunken, like we've spoken about kishmish, when we put them in water, they have swollen up. Okay, so because they have swollen up, which means they have taken up water. Okay, so that is nothing but the process of imbibition. ठीक है, जो we have studied, जिसमें हमने almond का example लिया था पहले, correct? Okay, so now going ahead, nobody is perfect. That's why pencils have erasers, right? So it means nothing but we are going to make mistakes. Pencil se bhi hum likhte hain, something goes wrong, we erase it and we go ahead, right? It doesn't stop there, right? So similarly, in life also, you make mistakes, but that does not mean any stop point for you. It's okay. You can still correct that mistake, learn from that mistake, rectify your mistake, don't repeat that mistake again, and move ahead. Okay, that's what life is all about. So yes, kids, now let's go ahead. We have studied about osmosis in our last session. Just a quick revision, what is happening in osmosis? What we have seen is osmosis is slightly different from diffusion. Diffusion में क्या होता है? Free movement होता है. But osmosis में this movement gets slightly restricted because of a semi-permeable membrane. ठीक है? It is the movement of particles, but it is via a semi-permeable membrane. So now this is the basics of osmosis जो हमने last class में देखा है. Now what we are going to see is two different types of osmosis. ठीक है, अब इसमें क्या हो रहा है? There is endosmosis and there is exosmosis. यहाँ पे देखो ये diagram में, these are two examples that they are given. First one is raisins वाला जो हमने activity में perform किया. Raisins पानी में डाल दिए. Next day when we are checking, they are swollen. ठीक है, इसका opposite करते हैं अभी. Let's take grapes, which are already swollen, right? Grapes are already swollen. So now let's put those grapes in water. अब grapes को जब आप देखोगे ना next day, they will shrink. मतलब they will appear shrink to you. ठीक है, तो ये exactly opposite हो रहा है, right? Raisins shrink रहते हैं, but वो swell हो जाते हैं, और जो grapes रहते हैं वो actually swollen रहते हैं, but shrink हो जाते हैं. So these two processes are nothing but endosmosis and exosmosis. Okay, let's see what is endosmosis. When a cell is placed in a hypotonic solution, the water moves inside a cell and swells. ठीक है, अभी what is hypotonic and hypertonic वो हम आगे discuss करेंगे. Water is moving inside the cell. Just remember this. Endosmosis में water is moving inside the cell and the cell is swelling up. ठीक है, जो raisins में हो रहा है. Then now what is exosmosis? When a cell is placed in a hypertonic solution, the water moves out of the cell. ठीक है, exosmosis में क्या हो रहा है? पानी अंदर से बाहर जा रहा है, मतलब बाहर निकल रहा है पानी, right? That means that cell is shrinking. Okay, that is exosmosis. Going ahead, what is osmotic pressure? Okay, what is osmotic pressure? Now let's see what is osmotic pressure. So now see यहाँ पे जो आपको level दिख रहा है, this first part of the diagram, you see have to be equal, correct? Now what is happening? Regular osmosis is taking place. Okay, there is movement of this particles via the semi-permeable membrane from low concentration to high concentration. Movement हो रहा है particles का. Now this movement which is happening, correct? ये जो U-shaped movement हो रहा है, that is somewhere or the else happening against the gravity. करेक्ट. तो ये जो मूवमेंट हो रहा है बिकॉज इट इज अगेंस्ट द ग्रेविटी, एक पॉइंट पे आने के बाद वी हैव टू स्टॉप इट, राइट. मतलब दैट वाटर कांट कीप फ्लोइंग अपवर्ड्स, राइट. इट नीड्स टू स्टॉप समवेयर. तो ये जो अगेंस्ट ग्रेविटी प्रेशर हो रहा है, इसको स्टॉप करने के लिए एक सर्टेन मिनिमम प्रेशर अप्लाई करना पड़ता है सो दैट ये पैसेज स्टॉप हो. ओके. So that pressure is known as osmotic pressure. Now see the definition. It is the minimum pressure that is exerted to prevent passage of pure solvent into solution when the two are separated by the semi-permeable membrane. ठीक है, is it clear? ये जो passage हो रहा है solute particles का या solvent particles का, उसको स्टॉप करने के लिए आफ्टर अ सर्टेन लिमिट. व्हाई? बिकॉज इट इज गोइंग अगेंस्ट ग्रेविटी. ठीक है. सो दैट प्रेशर इज नोन एज द ऑस्मोटिक प्रेशर. ओके. नाउ देयर इज समथिंग कॉल्ड एज टोनिसिटी. Okay, tonicity basically, let's see the definition first. It is the relative concentration of solution that determines the direction and extent of diffusion. ठीक है. डेफिनेशन स्लाइटली डिफिकल्ट लगेगा आपको, बट क्या याद रखना है, टोनिसिटी से रिलेटेड हम क्या देखते हैं, वी चेक द कॉन्सेंट्रेशन ऑफ द सॉल्यूशंस, ओके, व्हिच इज प्रेजेंट इनसाइड द सेल एंड आउटसाइड द सेल. ठीक है. तो इनसाइड एंड आउटसाइड का जो कॉन्सेंट्रेशन होता है सॉल्यूशन का, वो हम डिसाइड करते हैं बाय टोनिसिटी. ठीक है. यू विल अंडरस्टैंड टोनिसिटी लिटिल मोर इन डिटेल व्हेन वी सी 2-3 अदर टाइप्स. ओके. As you can see the diagram, we have 3 types of solutions. We have isotonic, hypertonic, and hypotonic. These are 3 different solutions. Now, isotonic, iso means what? Balance, equal, correct? So, the concentration is concentration of the solute, solvent, solution, everything is going to be equal inside and outside. That means when this concentration is equal, मतलब जो हमने फर्स्ट डायग्राम देखा था ऑस्मोटिक प्रेशर में, मतलब ये सिचुएशन होने वाला है ऐसा, जहां पे इक्वल है. ठीक है. तो इक्वल होने की वजह से ऑस्मोटिक प्रेशर तो होगा ही नहीं कुछ भी, ऑस्मोटिक प्रेशर नील होगा, इक्वल होगा. ठीक है. नाउ बिकॉज ऑस्मोटिक प्रेशर इज इक्वल, कॉन्सेंट्रेशन पूरा इक्वल है, तो ऑस्मोसिस होगा ही नहीं. ऑस्मोसिस क्यों होता है? इट हैपेंस व्हेन देयर आर 2 डिफरेंट कॉन्सेंट्रेशंस, करेक्ट, लो एंड हाई. बट यहाँ पे जब कॉन्सेंट्रेशन ही इक्वल है तो ऑस्मोसिस कैसे होगा, राइट? सो ऑस्मोसिस इज नॉट गोइंग टू हैपन बिकॉज द कॉन्सेंट्रेशंस आर इक्वल. ठीक है. आर वी क्लियर विद दिस? ओके. नाउ वी विल सी व्हाट इज hypotonic and hypertonic. Okay, let's see the definition of isotonic first. So 2 solutions having the same osmotic pressure across the semi-permeable membrane. ठीक है, as we discussed. Next thing, hypotonic. Now what is hypotonic? It refers to a solution that contains lower amount of solute as compared to the solute concentration in the other solution across the membrane. ठीक है. Again, now this is coming to a point where there is lower solute and higher solute in comparison with the 2 solutions. Hypertonic में, it is the one that has higher solute concentration outside the cell than inside. Opposite है दोनों, एक में outside ज्यादा है, एक में inside ज्यादा है. Now see, this is just the example. ये देखो आइसोटोनिक में देखोगे तो आइसोटोनिक में प्रेशर इक्वल रहता है, करेक्ट. यहाँ पे देखो ये सेल्स रेगुलर, यू कैन सी फील द सेल्स आर रेगुलर. सी हाइपोटोनिक सॉल्यूशन के सेल्स दे आर स्वोलन अप, करेक्ट. एंड नाउ सी हाइपरटोनिक के, सी हाइपोटोनिक में क्या हो रहा है, पानी अब्सॉर्ब हो रहा है. सी हियर यू कैन सी वाटर इज गोइंग इनसाइड. हाइपरटोनिक में वाटर इज गोइंग आउटसाइड, पानी बाहर निकल रहा है, इसलिए सेल्स श्रिंक हो रहे हैं. ठीक है. सो दैट इज अबाउट hypertonic, isotonic and hypotonic. Now we'll talk about turgidity. Last session we discussed about 2 terms, turgid and flaccid. ठीक है. तब I asked you all to keep in mind turgid means swollen, flaccid means shrunken. So here the definition of turgidity, it is the state of being turgid or swollen, especially due to high fluid content. ठीक है. It refers to the conditions of being bloated, distended or swollen. ठीक है. These 3 terms mean the same thing, that is swollen. Now, when we are talking about turgidity, we have to keep in mind 2 terms. Okay, kids, now please listen and understand this really nicely. Turgor pressure. ठीक है. We have a cell. Cell के अंदर कुछ contents हैं and we have a cell wall. ठीक है. जो सेल के कंटेंट्स होते हैं, when those contents exert pressure on the cell wall, that is known as turgor pressure. Okay, pressure exerted by the cell contents against the cell wall. ठीक है, इसका exactly opposite है cell wall pressure. Cell wall pressure में cell wall pressure exert करेगा on the contents of the cell. ठीक है, are we clear with this? See, you can see in this diagram as well. Here there is turgor pressure. ठीक है, टर्गो प्रेशर अंदर वाले कंटेंट्स बाहर प्रेशर एक्सर्ट कर रहे हैं, वॉल प्रेशर मतलब वॉल प्रेशर अंदर एक्सर्ट कर रही है. ठीक है, टर्गो प्रेशर, it is the pressure applied by the content of the wall against the cell wall. And cell wall pressure में क्या है? It is the pressure applied to the cell wall on the contents of the wall. ठीक है, on the contents of the cell. Going ahead, flaccidity. Flaccidity is nothing but shrinkage. That means it is the opposite of turgidity. Turgidity में अभी हमने देखा कि swelling हो रहा है, यहाँ पे shrinkage होगा. ये जो shrinkage होता है साइटोप्लाज्म का, that is nothing but plasmolysis. It is known as plasmolysis. What is lysis? Lysis means death. ठीक है, plasma. Plasma is present in the cytoplasm, right? So cytoplasm का death basically, cell is going to lose shape. प्लस साइटोप्लाज्म श्रिंक हो जाएगा, तो ये प्रोसेस को वी कॉल इट एज प्लाज्मोलिसिस. ठीक है, श्रिंकेज ऑफ द साइटोप्लाज्म अवे फ्रॉम द सेल वॉल. नाउ व्हाट इज डीप्लाज्मोलिसिस? व्हेन अ फ्लैसिड सेल रिटर्न्स बैक टू वाटर बिफोर डेथ, देन इट स्वेल्स. ठीक है, अभी सेल श्रिंक हो गया है, फ्लैसिड हो गया है मतलब श्रिंक हो गया है, बट ये श्रिंकन सेल जब वापस अगर पानी में आ जाए और वापस अगर स्वेल हो जाए, then that process is known as deplasmolysis. मतलब the cell is dead. Plasmolysis में death हो रहा है cell का, तो cell is dead. यहाँ cell is shrunken, but when that shrunken cell again swells up, that is known as deplasmolysis. Okay, opposite of plasmolysis. See, you can see it here. Turgid है, ये swollen है, और ये flaccid है, ये shrunken है. See how the cell has lost its shape. So this is again turgidity and flaccidity. Swelling, what are these structures? These structures are nothing but openings known as stomata. Okay, we will study about these things in detail in our next sessions, in our next chapters. So these are stomata, which are openings in the leaves and the parts of the plant. ठीक है, so ये जो opening है stomata का, this is happening because the cells are swelling up. These two are known as the guard cells, and these two guard cells are swelling up. ठीक है, and then when the water leaves, these cells are said to become flaccid. मतलब जो हमने देखा भी that they shrink. Okay, so this is flaccidity. Now this turgidity which is happening, ये जो cells swell हो रहे हैं, इसका use क्या है? First thing is it provides rigidity to the soft tissues. Now what are the soft tissues of the plant? Something like the leaves, right? So it provides rigidity because पानी अंदर जाता है, cell swell हो जाते हैं, तो they become hard, and that hardness provides some rigidity to the soft tissues. Okay, second one is it helps to push through the hard ground. अभी जो छोटे छोटे seedlings होते हैं which are inside the soil, ठीक है, उनको soil में से, यहाँ जब hard ground होता है, वहां से बाहर आने के लिए, when these cells are swelling, now they are becoming big. When they become big, they have good pressure, so they can use that and push the seedlings upwards through the hard ground. Okay, then they help the root cells to build up root pressure. Okay, it helps opening and closing of the stomata. What we have just seen last slide, this is the opening and closing of the stomata, and it helps the turgor movement. Okay, in the mimosa plant. Now, I'm sure you must have heard about the plant, which is also known as touch me not, right? When you touch that plant, the leaves will close. When you leave that, leaves will open. जब भी आप उसको टच करोगे ना, लीव्स क्लोज हो जाते हैं. That is known as the mimosa plant scientifically. It is also known as the touch me not plant. ठीक है, तो ये जो swelling हो रहा है ना cells का, this facilitates this turgor movement of opening and closing of the leaves of the mimosa plant, for example. Okay, children, are we clear with these uses of turgidity? Yes. Okay, so now today everything that we have seen in our lecture is all about osmosis. We saw about osmosis again. We saw what is osmotic pressure. We saw endosmosis, exosmosis. Then we saw 3 types of solutions based on the tonicity. Then we saw what is turgidity, what is flaccidity, what are the uses of turgidity. Correct? These are all important terminologies before we proceed to our next session. Okay? Now, again, coming back to the activity question. This you're going to tell me, think little bit slightly and tell me what the answer is going to be. Put the answers in the comments. Salting of meat is a method of killing bacteria by the process of dash. ठीक है, हम मीट को सॉल्ट करके रखते हैं, राइट? आई एम श्योर यू ऑल नो अबाउट दिस. वी इवन पुट सॉल्ट इन द पिकल्स फॉर प्रिजर्विंग इट, राइट? तो ये प्रोसेसेस क्यों होते हैं और ये किसकी वजह से होते हैं? व्हाई? बिकॉज ऑफ व्हिच प्रोसेस? योर ऑप्शंस यू हैव प्लाज्मोलिसिस, टी प्लाज्मोलिसिस, टर्गो प्रेशर और नन. ठीक है, यू आर गोइंग टू गिव मी द आंसर इन द नेक्स्ट क्लास एंड वील डिस्कस दिस अहेड. So yes, I think we have reached the end of today's session. So we've discussed all the basic terminologies. So kids, please go back and go through these terminologies. Okay. And I will see you next class. Bye-bye.